So Yehuda is a professor at the Washington University in St. Louis in America. He has his own research group there where he's interested in how genetics can influence behavior. For this particular project, he's brought in the microbiome as well, working with someone from his research group, I think that she was a graduate student, right? Cassandra Vernier, looking at how the microbiome and the host genomics can influence behavior in honeybees, which I'm super excited to hear about honeybees. So off you go.
Well, thank you guys for inviting me. And thank you for not scheduling it too early. So it's manageable. Yeah, I have to admit right up front that I'm a bit intimidated talking about this topic to people who know what they're usually talking about when they talk about hologenomes and microbiomes, because I feel a bit of an accidental tourist in this field. But I really got into it because, well, obviously you guys here, so you all agree that this is a fascinating area of research, the interaction between animal hosts and their microbiomes. And I come from a direction of really behavioral genetics, trying to understand how behaviors are encoded in genomes. And the whole, I know maybe it seemed trivial for a lot of you, but for me to start thinking about the mechanistically how, in fact, it's not just the host that we need to look at, but it's in fact this hologenome, which again is a concept that even the term itself was something that I introduced to in the last couple of years. I didn't even know it existed until we started working on these questions, or at least I kind of knew about it peripherally, but you know, in a naive way, I was very, the way I was thinking about the questions that we work on intellectually was always very sort of animal genome centric. So yeah, so we are, it's pretty early. We published a couple of papers, but it's really, I hope that I'll get some engagement for you guys and get some good input, and we're looking forward to keep figuring out this really what I think is fascinating. All right. I always give too long of an introduction. So let me tell you a little bit about the way we think about it. So we came to these problems really from the direction, again, of the animal, and in this case, it's going to be bees. It sort of evolved from discussions about just kind of the concept in general of self-recognition and group relatedness, which is something that in social insects is a very important aspect of their biology. And so we've been really sort of trying to figure out like ways to tackle some of this sort of a biological aspects of how group relatedness is regulated and sensed, and what kind of signals and neural pathways are involved in it. But it's a very, conceptually, it's a very broad, right, biological sort of problems that we find at all different levels, right? I mean, if you think about immunity, if you think about like an organismal biology, like, you know, for example, brood parasites, right? I mean, so there's a lot of examples of how relatedness and abuse of regulation of relatedness or pathways that regulate recognition and response to relatedness signals are used in nature. And mate choice, which is something that we've been working on for, I could say probably originally triggered some of these thoughts, is about decisions about who to mate with. And I think this is probably photoshopped. I just found that image online somewhere. But you can imagine how devastating it would be when you really are not good at making these decisions, right? So social insects, and I'm going to use honeybees, European honeybee, which is the most common social insects. You know, it's a farm animal, right? It's a domesticated organism, and almost everybody's familiar with bees because they're everywhere. The problem is actually extremely complicated because they live in these huge colonies. I mean, in maintained colonies, you know, those hive boxes that you see in fields, you can have sometimes up to 40, 50,000 individuals that are all representing one family, right? One colony. And it's kind of crazy to understand, how do they know that they all belong? And it's important to know who belongs, because their whole shtick is to basically get resources from the environment and concentrating in one spot. I mean, this is why bears go after hives. They actually not usually go after the honey, they go after larvae because it's a protein source. But nevertheless, I mean, there's a lot of energy and protein all concentrated in one little spot. And if you know how to find it, it's easy, right? And so they have to defend themselves constantly. And most of the time they have to defend themselves not from bears, but from other bees. Because you can imagine it's so much easier to go and just invade a hive and get their honey rather than go and look for flowers and collect pollen and all these things. And if you can just walk in and take it, it would be much simpler. Yet it doesn't happen very often, or it does. When it does happen, there's a lot of conflict. So how do they know? Obviously, visually, there's no way. Well, maybe they could, but it would be hard to imagine how a small brain like that can remember and recognize 40,000 colony mates. So so in reality, we know for a long time, we knew for a long time, and a lot of it comes from actually from farming practices and from beekeepers, is that it's most likely chemosensation. We knew for a long time that bees, and it's probably true for a lot of other species of social insects, ants and social wasps and so on, they really sort of somehow recognize certain chemical cues that are pheromones that are on the cuticle on the outside of the animal. And since they regulate who comes and who goes at the entrance of these colonies, they can actually act. There are certain individuals that in bees, we call them guards, can actually regulate who is allowed to go in or out. The rest of the talk, like most of the data I'm going to show you, is actually work of Cassie Vernier, who recently graduated from my lab. She's now a postdoctoral fellow at the University of Illinois. And she, when she joined my lab, she really was interested in social insects. For those of you who don't know me, in my lab, actually, most of the work we are doing nowadays is with Drosophila melanogaster, the vinegar fly, and related species. But thanks to Cassie, I kind of got back to my roots with bees as well. So mechanistically, without getting into a lot of the biology of bees, but if you didn't know, I mean, these colonies of honeybees, and it's true for, like I said, for other social insects as well, is that most of the individuals that I showed you in the picture, the very busy picture with all these bees, are workers, which are sterile females. In honeybees, usually there's a single reproductive individual, one what we call queen, which is basically, she's not really a queen, she's just an egg-laying machine. And so she's like the germline of this sort of, some people like to call it the superorganism. And majority of the workers act as like somatic tissues of that superorganism, in the sense that they do all kind of different things. Right, they take care of brood, some of them forage, some of them act as guards. And in bees, again, this process is what we call age-dependent polyethism or behavioral development. So all bees usually do most of the tasks, they just do it at different ages. And it's a pretty tight regulated process, circulated through hormonal and various neuroendocrine pathways. And so there's a lot of work. Bees have been used for this as a model to understand behavioral plasticity and how bees sort of make the decisions to switch between tasks. And part of this whole complex regulation of who's doing what is the part that I started to talk with, which is recognizing who belongs and who doesn't. And when we kind of try to understand what actually regulates it, because all the members of each single colony are all actually related sisters, just because of the way that sex determination and reproduction works in these organisms, the simplest way to explain it is that they're all genetically related. So maybe there are some alleles in each colony that represent some sort of a signal, right? And they all sort of develop to recognize it at the colony level. And so if you don't belong, if you're genetically a little bit more distant than members, they can sense it and they reject you, right? However, multiple studies and hundreds of years of beekeeping tell us that this cannot be really the mechanism. And I'll show you some data from us and others that support that. So the fact that it's probably genetically relatedness is not what regulates this signal. It has been recognized already in the 70s, and people tried very hard to come up with all kinds of models to explain this phenomena that seems to follow the rules of genetics, yet is not, or kind of blind to genetics in some way. And one of the most common models is the Gestalt model, and the idea is that the signal is, instead of being determined genetically, it's an emergent stochastic signal. It's somehow, these animals, they forage in the environment, they bring all kind of things into the hive. There's all kind of oils and fatty acids and mix of pheromones and all these different chemicals somehow generate some sort of a signal that stochastically is unique to that group. And they all, because they live in this dense environment, they share these, they rob each other, they touch each other, and somehow it spreads in the colony. And magically, they all learn it, even though it's stochastic. There was some empirical evidence that it might work in this way, but hopefully I'll convince you that it's probably not, that's not how it works. And that's where the microbiome will come into play later. Okay, so one of the reasons why the Gestalt model was sort of, it made sense mechanistically to a certain degree is that, so this is a picture of a bee, a brand new bee, as she emerges from her cells. So the pupae are in these cells, and you can see some of them are still sealed, and you know that she's new because she's all these like nice kind of unbrushed hairs all over her. They're very cute. They can't sting, so we really like them, and they can't fly either. So it's easy to mark them and things like that when they're just babies like that. The one thing is that we also know is that when they come out, they barely have any really pheromones on them. Like when if you measure these with a variety of different methods, you can show that they're kind of like blank slate. And so it's been known that whatever becomes the sort of the unique pheromone of any member of that colony, somehow it's a delayed trait. It's somehow related to, as the bee mature, the signal matures as well. Here are some data to show that it's probably not genetics. This is from '99 by Dons and Ratniks, where they did just some simple cross-fostering experiments where they looked for acceptance by the guard. So you basically take bees and you place them at the entrance to the hive and see how the bees react to that bee. And so acceptance means that they allow the bee to basically walk into the hive versus rejecting it, so preventing either stinging it and killing it or just kind of pushing it away. And you can see that in general, whether they were by related means genetically related or unrelated, because if you take these very, very brand new bees and you dump them into a colony, even if they're not completely unrelated to all the other bees in that hive, if you introduce them early enough, they seem to acquire that sort of whatever the signal is. And so you can see that in general, it doesn't matter if they're related or unrelated. If they're nestmates, they'll be accepted. If they're not nestmates, even if they are related, they'll be rejected or accepted at a much lower rate. So again, suggesting that really genetics by itself is not enough to explain the phenomena. So just to kind of summarize the Gestalt model, which originally was proposed by Crozier and Dix in '79, is that somehow, even if we have a group of organisms that are all a little bit different in terms of their innate signal, the Gestalt is like somehow this process, this environmental homogenization, I always have a hard time with that word, sort of generates some sort of a more average signal that is shared by everybody, and somehow they all learn it, and they know when somebody doesn't have this purple, fuchsia, whatever. I didn't pay attention to when I started, so I don't know, in terms of keeping time, hopefully somebody can help me with that. And so the idea is that each hive, right, develop its own Gestalt sort of signal, and that's how they know who's you know, blue, who's pink, who's whatever. So the one thing that Cassie did in her first thesis chapter was to really revisit the whole idea that there is Gestalt. And so, and what to summarize the whole paper in one figure is that basically we did not see any evidence that there is Gestalt. So what in this specific case, I'm showing you data of the CHC, of the cuticular hydrocarbon profile, which is what the bees use as pheromones. So it's a complex blend of different hydrocarbons on the cuticle, kind of a waxy compound. And basically she sampled bees that were like she took day-old bees, like those bees that just emerged, and introduced them back into their own colony or to completely unrelated colony, and looked at their profile over time. And what you can see is that there's a very strong age-dependent changes in quantitative and qualitative changes in their profiles. And this is a discriminant analysis, so it's kind of reducing the complex data to a two-dimensional space. But what you can see that it didn't matter if in their own colony or in unrelated colony, there was a very strong age-dependent changes. So this really suggested that there, or goes against the idea of Gestalt, because it seems that age is much stronger than any, you know, it doesn't matter that you're in this hive with all these bees, like you would look, if you're 7 days old, you'll smell differently from a bee that is 18 days old, which is usually when they forage, or, you know, this is what they look like. You can see how tight they are in the first day, and then it's a little bit, the clouds get a little bit more sparse, but they're still clustered. This is really suggesting that there is a regulated process. It's not just like everybody come in and it's some sort of an average, everybody smells the same, because otherwise age would not have had such a strong effect. Hopefully that makes sense. And this is what behavior look like. So this bee here with green is the introduced bee, and you can see already that there's a guard that is very interested in her. And you can see there are returning foragers. They come in, how they ignore this drama here. But I'm not going to show you. I mean, basically, this guard is going to push that. You can see now she actually drags that unrelated bee. And so it's not true that they always like extremely aggressive in terms of killing the other bees. Most of the time, they'll drag them out and just let them fly away. Okay. So it's a quantifiable behavior that you can actually study in the field. So we felt that both of these models, of course, the first one we knew already, and that was determined by others before us, but even the Gestalt seems to really not be a good model to explain what's going on. And we came up with this ontogeny model with the idea that this is actually a physiological process. It's a developmental regulation, the signal, and again, I'm trying to summarize a lot of data before I start talking about the microbiome, but the idea was that it's really the signal itself is actually not Gestalt across all colony members. It's really unique to foragers, and the whole interaction is unique to this geographical or the specific space in the hive, which is the entrance, where guards and foragers interact. All other bees, in fact, are completely sort of agnostic to these signals. They don't care if you belong or don't belong. It's the guards and the foragers that's where the interaction is happening. And so we decided to focus on that area and to try and understand, because we know foragers are the oldest bees. That's what they develop towards. So they start as a nurse, they become foragers. And our data, again, suggested that the CHC profile sort of develops, and where the most divergence is happening in terms of the signal itself across colonies happens at the foraging level. So there is like a development of a signal, and somehow the signal at some point becomes unique to the colony at the level of foragers, okay? And that's where we started with trying to understand, okay, how this signal is developing, what is the actual physiological process that drive it. So it seems that it's innate, like, so it seems that it develops as the bee sort of ages and switches between tasks, yet it's seemed to be completely blind to the actual genetic or genetic background of each individual, because it seems the colony is a much stronger factor in terms of determining the specificity of the signal across different colonies, which is really puzzling. How can you have something that is innate and developmentally regulated, yet not seems to be determined by the genetics? But that's, again, was because of our bias and looking at the genetics of the host. So hopefully I'll convince you that it is genetic, but it's genetic of microorganisms that really determine this. And this is where we evolve the hypothesis to what we call now the shared gut hypothesis. And maybe we should, in retrospect, call it the hologenome hypothesis. And this is where sort of our thoughts start evolving, we thought we have to look outside the genome of the bee to try and solve this puzzle, and not really solve, but at least start solving it. And we've known, as I'm sure many of you know for a long time, is that in fact the role of microbial symbionts in social behaviors of animals has been recognized already in the 70s. For example, Indian mongoose, we know that individual recognition depends on bacteria in some of their pheromone glands. Group recognition in hyenas seems to be dependent on bacteria. We knew from termites, again, it's not a social insect, it is not a hymenopteron, it's actually like a social cockroach, actually. Clearly microorganisms, a lot of them are eukaryotic microorganisms in their guts are important for this. And mate preferences in fruit flies, also the microbiome has been implicated in that. Against hinting that evolution took this association between the animal host and bacteria and sort of allowed this symbiotic system to evolve in a way that we sort of from the outside perceive it as a pretty robust mechanism that regulates how animals know at the chemosensation level who's who, okay? Which could work, right, for recognizing relatedness as well. So what sort of like kind of remodeling the way we thought about it is that, and this is the kind of an oversimplified cartoon that explains it. What we mean by the shared gut is that, you know, there are certain environmental inputs, environmental factors, evolutionary factors that that shape, you know, in this case the honeybee, and somehow via direct or indirect effect allow this emergence of a colony signature, and in different colonies this emergence of this shared gut factor or system or whatever you want to call it, or hologenome perspective, is what defines what will end up being the unique feature. So it's the host plus whatever is in their gut, which they can share, yet it is sensitive to the developmental, the ontogeny, so it sort of fits a lot of the bits and pieces of what people have described in this phenomena, right? In terms of how it develops and how it emerges in a sort of specific environment independent of the genetics of the host. And again, if I'm being vague, hopefully it will be clear when I show you the few more data slides. How am I doing with time?
So we're pretty much at question time, so you can keep talking because it's super interesting, but the more you talk, the less questions.
Sorry. Okay, so just to make it, just to hopefully I'll convince you that we're probably on the right track, is that first of all, what Cassie did is she took foragers from 3 different colonies. Remember I told you it's the foragers that are where the signal is. And when you analyze both their microbiomes and CHEs, which stands for the profile of their pheromones, what you can see is that in fact for both factors, which are again complex, right? Complex systems. The microbiomes is a bunch of different bacteria. The pheromone profile is a bunch of different chemicals on the cuticle. You can see that each forager group had their own sort of signatures. So in some way they're associated, right? It doesn't prove any causality, but at least it suggests that it's possible, right? When she did cross-fostering experiments, so again, cross-fostering is you take bees that emerge in one colony and you transplant them as young bees in another colony. So the colors here, the red is from colony 1 and the blue is from, sorry, the circles are colony 1, the squares are colony 2, and the color is which colony they were raised in, right? So there are 2 independent colonies here. And what you can see that overall, it seems that there are effects of, if you look at the statistics, effects of both your source colony, your host colony. So it seems like, at least in these experiments, in one case, for example, when bees were introduced to colony 1, really it didn't matter where they came from. They looked very similar. You can see that the letters here represent the statistical difference. But in bees from colony 1, when bees were raised in colony 1 from either 1 or 2, they were still different. So it again seemed to have some context specific. So in some cases, really the only thing that matters is the host colony. Doesn't matter where you came from. In other cases, it seemed like there was some mixed factors involved in it, in what determines your at least your microbiome at the time where we tested these bees. But to directly test it, we decided to do manipulations. And again, I'm not going to show you all this is all now published. It was published relatively recently in 2020. When you raise bees in small cages with bees from different sources, because we know that bees, one way that they gain their microbiomes, for example, is by trophallaxis. They exchange poop and food from both ends, and sort of they kind of similar to how newborn babies, right? The whole idea is that breastfeeding is how they get their first microbiomes in their guts. What you can see is that when bees from the same source were raised with bees from different colonies, they end up with different microbiomes. So clearly you can end up diverging the microbiome, and it ends up having also different cuticular hydrocarbons. So again, black and red are bees that were raised with bees from the different colonies. But if you first treat the bees that you house the young bees with antibiotics, you lose that. So again, suggesting that really the pheromone profile becomes kind of random, or at least it doesn't cluster by colonies, suggesting that really the older bees that were housed with the young bees, their microbiomes is important for determining what the pheromone profiles of the young bees will look like, if that makes sense. And I'll finish by something that, I don't know, some of you might think is radical. I actually never thought it's that radical, but a lot of people in the field of social insects don't like this idea, but I'm going to say it anyways, which is, and it's a little bit of a can of worms to get into kin selection and all that stuff, for any of you who are familiar with that field. But the one thing about social insects, which is fascinating, is that something in Hymenoptera, so Hymenoptera is the order of insects, includes bees, ants, social wasps, includes both, right, social animals that are eusocial, so they're like honeybees, ants, and so on and so forth, as well as, like I said, multiple lineages that are not. And what's interesting is that it's, by resolving the phylogenetic relationship between all these species, it's clear that this eusociality trait emerged multiple times independently. It wasn't just like, even though it's a very complex, very large, and very ecologically successful order of insects, it seems that something in the genomes of these Hymenopteran, at least that's the common theme right now, is sort of making them sensitive to whatever evolutionary forces that push them to evolve eusocial, like the colonial type lifestyle. Okay, we know that the ancestral species are always solitary, and so it happened, like I said, multiple times independently. And often when it happens, there are very, very similar traits that evolve, like kin select, like nestmate recognition, so on and forth, and so forth. And a lot of the explanations are based on kin selection, which again, if you're not familiar with the whole theory, but the idea is that because the bees are all genetically related to each other, it's a driving force. I just showed you, for example, that some traits, that genetic relatedness has nothing to do with it. But I'll finish by giving you my idea. I mean, it's not my idea completely, but it's like some sort of sort of a thought exercise is that what if in fact this has nothing to do with the host, but this is all a microbiome derived trait. So what if, we have a complex society of individuals that all have different microbiomes, and each one has its own system in their gut, although it could be in other organs as well. If the microbiome sort of pushes colonial expansion, in fact, it serves the microbiome, because you now, instead of having just one animal as your niche, one specific individual, you have a whole colony. So your fitness and it could be that maybe there's one key bacterium in there that evolved very early in the divergence of this whole group of insects, that is, whatever the interaction of that specific bacterium with the host is the main things that pushed. And one way that this is testable is when we start figuring out really what are the hologenomes across many different species in this group, it will be interesting to see if there are some clear signatures in the hologenome that may explain which lineages ended up showing this eusocial trait, sort of the colonial expansion. And of course, then they all have to protect each other. So this is where the nestmate recognition comes, because the bees and their microbiomes want to protect themselves from the bees and microbiomes that are unrelated. And we have actually some evidence to it, but I'm not going to have time to go over it. And this is Cassie and the rest of people in the group, and I'll stop here. And I'm sorry I went longer.
I didn't want to stop you because it's just so interesting. I think we have time for one quick question to keep moving.
Sorry, guys.
No, that's okay. Tom has put a question in the chat, and he is the head of the center where I work. However, I'm going to ask my question because I'm the one that has the microphone. It seems
and feel
to me
free
that,
to email me and stuff if you have other questions.
And I will send you the question that he wrote as well. It seems to me that if it is driven by the microbiome, does that make it vulnerable? Because everyone, of course, honeybees, the problem is all the bees are dying and we hear about it. Is this something that could be really disrupted by something in the environment that upsets the microbiome? Or
there are people who look at this not from the perspective of the evolution of eusociality, but from just the perspective of bee health, because it's an economically important species. So there's a lot of work that, yes, if you disrupt the exposure to antibiotics in the environment or all kind of pathogens and things like that, and one of the ideas for all the disappearances is that there's multiple, the multiple assailant kind of hypothesis, that it's kind of like a perfect storm. Multiple things end up reducing the fitness to a point where the bees just can't take it anymore, and pathogen load and sort of the replacement of the healthy microbiome by pathogens, things like that, is part of this. So I think that's the fact that the microbiome is important for all animal hosts. I mean, I'm yet to wait for somebody find animal that you can keep completely bacteria free, and these remain healthy animals, right? I mean, we know that the symbionts are important for the host. I mean, we have no existence without them. I mean, I'm pretty convinced about that. So it's true for bees as well.
So since Yehuda has to actually leave after the talk, I will also ask Tom's question. It is a good question. He's basically, I'm going to summarize it a little bit. I'm sorry, Tom, but he's basically saying how much you think the genetics of the host plays a role. So for example, can all colonies within a species or population able to pick up the same microbes or produce the same chemicals? Or do you think that there are maybe subsets where, because the genetics of the host is too divergent, then it's feeding back the other way?
Yeah, so I mean, I don't have a direct answer for it. I mean, it's something we are starting to look at, but I have zero doubts that it's an interaction. It's not, I mean, when I say completely blind to genetics, so the signal specifically for nestmate recognition seems to be more, the microbiome genetics is more important than the host, but this is just one dimension of that interaction. I mean, there's a lot of other things, and I have zero doubts that if you start looking at allelic contribution to fitness, and you look at it as a whole genome, of course there's going to be, there's no way that it's not involved constant interaction. And you can imagine single bacteria with certain allelic combination would be a much better partner to some hosts and not optimal to others. And you assume that there's a process that always, right? I mean, it's so I have zero doubts that for a lot of traits that we'll find much stronger interactions. That hopefully I'm answering the question. But that's
and FindingPheno's whole project is about integrating the data from the host and the microbiome. So I'm very happy to hear you say that.
Yeah, and I'm very interested in. I'll email you guys later because I was reading last night about what you guys do, and I think I need to talk to you more and learn a lot about because it will
definitely go both ways.